we have been talking about viruses. So I think we left off, we were examining how the different viruses that belong to the different classifications, they work their replication or synthesis process a little differently, depending on what kind of nucleic acid they have to begin with. And also, when you think about the host cell, they have to essentially, a lot of them use almost everything that the host cell machinery provides. Sometimes they bring their own enzymes to help. But, when you think about the typical process, if you're going to make proteins, and the virus needs to make a lot of its own proteins, you have to go through the route of, you start with double-stranded DNA, and then a strand of that will be used to make messenger RNA off of it. So that's the transcription step. And then a different polymerase will be used to take that messenger RNA, the ribosomes, all of that, get translated into protein. So the virus has to use that. And then it also has to make copies of its nucleic acid, whatever kind it has. So it's got essentially two main goals to accomplish. And so it really does depend on what they bring with them in terms of their nucleic acid. So if it's a double-stranded DNA virus, that's probably the most straightforward case because it's pretty easy for them to immediately take the host cell machinery hostage, so to speak, and make messenger RNA off its double-stranded DNA. One of the strands will be used for that. And then it can go ahead and make protein off of that messenger RNA. So that's straightforward. The single-stranded DNA viruses, what they do first is they get their single-stranded into a double-stranded state. And so they have to make the complementary copy so that they can get into the double-stranded state. Then they're good to go. Then they can go ahead and use one strand of that to make their messenger RNA and then the protein. Then single-stranded RNA viruses that are in the plus configuration. So there's positive strand RNA viruses. And if it's in the positive sense, they can actually use the word sense. If it's in the positive sense, then that means it can be directly used to make proteins off of it. They can use it as messenger RNA, do the translation step, make protein. And so typically these viruses have on their RNA the genes necessary to make RNA polymerase for what they need to do. And then they'll be able to use that RNA polymerase to make a bunch of copies of the messenger RNA minus. So that's the complementary strand to the plus strand. We always talk about plus and minus being complementary when we get into nucleic acid. So this step is good because basically when they use that RNA polymerase, they're going to, well, actually what they do is they make this negative messenger RNA and then they make a bunch of positive RNA out of that. Right? So it's a way for them to make sure that their single-stranded RNA plus, they have plenty of that nucleic acid made. They have to make more of that. They have to make a bunch of proteins. And so if they can code for the RNA polymerase, that sets them up to make a bunch of copies of their nucleic acid plus RNA version. And then they can package that in and make a complete viral particle. So they're doing two things. They're making sure that their proteins are getting made, but they also have to make copies of their particular type nucleic acid. Right? And so that's a little different. If they are the so-called minus strand type of viral particle, where they have a minus RNA and it's single-stranded, or if they have double-stranded RNA, what they do is they bring an RNA polymerase with them, and of course their nucleic acid. They get into the host cell, and they are going to use that RNA polymerase to make essentially the positive negative strands provided. And that's what they're going to use then to make protein. So remember, if it's the positive RNA version, that's the strand that, yep, you can use that. You can use that to make protein. Okay. So this single-stranded RNA minus, and then the double-stranded RNA, they'll take the minus strand and use the RNA polymerase to make the plus strand off of that. And so it's just this game of they got to make sure that they can make enough of their own nucleic acid, and then they also have to make their own proteins. The retroviruses, those are the ones that have that very special reverse transcriptase, where they actually will be able to go backwards, so to speak, in the sense that they can make DNA off of RNA. Typically, we always teach that it's DNA to RNA to protein. That's the way it is. Of course, there's always an exception in science. And so with the reverse transcriptase, you can take an RNA virus, single-stranded RNA virus. You can use reverse transcriptase to do that first step, make a single-stranded DNA off of RNA. Very peculiar. Then the virus has to make a complementary copy to get it into the double-stranded form, and then it's good to go. Then once it has the double-stranded DNA, it can go ahead and use the host machinery, make messenger RNA, and then protein off of that. So and then there's some other even weirder examples that I'm not going to get into, but this image right here is pretty much talking about what we just talked about, bulletin points, right? And so this is a visual of what we just talked about. If this is easier for you to work through it, this is another option. So here there's some, yep, the double-stranded DNA, we immediately take the host machinery and work with that to make the messenger RNA. And you're not showing the protein step, but once you get the messenger RNA, you can make your protein off of that. Okay. And then, so this I believe is from the current edition, and then these are actually not from your book, but I put these in here too. If this is a different way for you to look at it, that helps you work through it. That's available too. Okay. But those are basically saying the same exact thing as the other image I showed. Okay, so this is all happening during the replication or the synthesis stage. And it's just this process where they end up with a bunch of proteins floating around in the host cell, a bunch of nucleic acid floating around. And so it's all bits and pieces. They're not good to go yet because it's like the assembly line hasn't fully assembled everything. And so those proteins, of course, remember they can be both the structural proteins used to make the viral particle itself, but then some viruses make special enzymes that they carry with them. And so there's both types. And then, okay, so all of this is floating around. So now I got to put it all together. So that's the maturation or assembly phase. And it's just getting all those bits and pieces put into a coherent, intact viral particle. And then the last step is release. And depending on the virus and the host cell, you will see that typically the host cell lyses open, but not all. So I'll show you some. So the release stage is after they've made their copies, and it depends on the virus, like how many copies per cell of the viral particle can be made, and it varies. It can be 10, 100, 1,000. It just depends on the host and the particular virus. So no matter which viral particle you're looking at, you have to get, the fully functioning viral particle all put together. And you can see, like with these double-stranded DNA particles here, very complex in their and different parts for sure. Very funky looking. And then you have some more simple phages as well. So it just depends. Here they're trying to show the idea visually looking at from, step one, which is attachment. Far left, you're seeing that, where you have the funky looking T4 attaching to the wrong one, attaching. And then they're trying to show the step-by-step process of getting the replication, assembly. And in this particular case, they're showing it's 25 minutes to get all the way to lysis. This particular T4, it has 25 different proteins that have to be made. And then many copies of some of those have to be made. Plus, they have to make a bunch of copies of the double-stranded DNA and get that all put together. So fairly complex. Okay, so then there's some phages that have been studied that are temperate. And we've actually gone over this already when we looked back, when we were looking at gene transfer mechanisms and some of the peculiarities, I guess. So when you think about the lysogenic pathway, that is where you have the double-stranded DNA of the virus getting pasted into the host DNA. We've talked about this. And then that stretch of viral DNA can just stay there. And it can stay there for a fair bit of time. In animals, it can stay in the host cell for years, depending on the virus. And so effectively it's hiding in the host cell. The host cell continues to live its life. It's usually okay. And that virus is just has its DNA hanging in there, but it's not making more copies of itself yet. So they think, at least in the cases they've studied in the lab, they think there's a repressor protein that the phage itself has that is keeping that phage DNA pasted in and securely in the host DNA. It's still kind of mysterious how this happens. And then basically when they're studying this in the lab, what they end up doing is once they get this step done where they can show that, oh yeah, E. coli, it has a big DNA in its own DNA, then what they do is they take that E. coli and they stress it. And they usually hit them with UV light, which you did the other last week in lab, or they'll apply a chemical that mutates the DNA. And that's enough of a stressor to trigger the viral DNA to cut out of the DNA of host bacteria. And typically when that happens, then when the virus is free floating in the host, it's cut out of the DNA of the host, and it's just floating around, that's when it starts to go into the very active replication phase. So we call it the lytic pathway, because then it starts going, okay, I'm going to make more of me, more of my DNA, more of my proteins, and then I'm going to bust out of this cell and go find another E. coli to infect. And so it's a fairly well-studied phenomenon. So I think you saw this already, where they're showing the two different pathways, and the darker green is the viral DNA, and they're showing it getting pasted in. And so when it's pasted in, I do want you to remember that term again, prophage. Okay, prophage. That just, if someone says, oh yeah, I know this E. coli has a prophage in it, what they mean is they know that there's some viral DNA pasted into E. coli's DNA. So then that's the prophage state, prophage. And then you can see that if this cell decides to go through cell replication, and there's two progeny cells, through binary fission, it just kind of doubles all of its content, makes a copy of its DNA, and splits in two. So each one of those progeny cells also has the prophage in it. So it's a very passive way for the virus to spread through a population of cells just by keeping its DNA pasted in like that, okay? And then you can see, if you take, it looks like they're suggesting this cell down here got stressed in some way. So induction, they just mean somehow there's some kind of trigger that got that DNA to cut out from the host DNA, and now it's taking that cell hostage and making more viral particles. So lysogenic pathway, and then it can kick into the lytic pathway, okay? This is another example of that from, I think this is more from your current version of the book with more words on it for studying. So this is a good one to look at for studying, but it's basically saying the same thing. So here, this one. So here with this example, they're calling it a slow release replication cycle. So this is pretty special because what they're trying to show here is that you can get this the virus taking the cell hostage, but it's not killing the cell when it leaves the cell. And it's often, they tend to see this with these so-called filamentous shape viral particles or phages. And so it just seems that when they exit out, maybe it's just because of their small, very, very small, slender size, they can kind of sneak out of the host cell. And as long as they're not doing it all at once, they can sneak out and they can keep that host cell alive. And then they're showing that the bacteria is healthy enough to just keep doing its thing, which in this case, it's replicating itself. And so they know that that can happen. So far as I know, it's like I said, with the really skinny, tiny little viral phages, where it seems like their shape helps with keeping the host cell intact. Okay. Animal cells, which they're the ones that cause us issues, or can. Those are the ones that when you look at them and look at what they're composed of, they often do the trick of they capture some of the host cell membrane. And it can be, well, it can be the cell membrane, but it might be an internal membrane. It might be part of the nuclear membrane, something like that. But it's host membrane material and it gets wrapped around itself. And so they tend to do that. And then it's a slower process because once they get into the cell, everything's all not just right there. Like with a bacteria, everything's right there, just floating around in the cytoplasm. But with an animal cell, you got all kinds of organelles with their own membranes. And so you have to think about all the logistics of not only getting in the cell, but then they have to get to the right parts of the cell to do the different stages of their transcription and translation. And so they have to make it to the nucleus of the host in order to do the DNA to RNA process and to do their nucleic acid replication. And then they have to get their RNA out of the nucleus and they have to get into the cytoplasm so that it can be used. It can, find a ribosome and start working through the RNA to protein step. So it's a longer process. It's more complicated. And so when you look at their replication, attachment to lysis timeline, it is longer for animal cells and the viruses that infect them. So animal viruses, there's lots of different animal viruses. Some are fairly benign and some are deadly. And so you're seeing some variety here of morphologies. Coronavirus is in there too, retrovirus. So just some different shapes and sizes. They're trying to do relative sizes here to show you the difference. So they often use this phrase, tissue tropism. So basically what that means is that you will see that animal viruses have very specific cells and tissues that they can attach to, and they can't attach to the other ones. And so it's the specificity, and it really has to do with the cell surface receptors that host cell has, whether or not the virus is going to be successful. And so it can be broad versus narrow. So Ebola, unfortunately, has a broad tropism, which means it has adapted well enough where it can infect, it can attach to many different receptors, which is not good. That's not what we want to hear in such a deadly virus. That's why it is so deadly. Within 72 hours of infecting a human, it can kill them. So papillomavirus is very specific. It only works on particular epithelial tissues. And so it just depends on the virus, how we're going to see that play out. Animal viruses, they tend to use the word virions. They enter the cell as virions. What that means is it's not just the nucleic acid that's getting in, it's essentially the whole viral particle that's getting in. And it's usually through endocytosis. And you have this like merging of the viral envelope with the cell membrane, and it kind of dumps the rest of the virus into the cell. Then, so if it's the entire viral particle getting in, it's got to break open. And the fancy word for that is uncoating. It has to uncoat. So all that capsid, those protein capsids have to break apart well enough to get the nucleic acid out. And so with animal viruses, with the DNA viruses, they are pretty good at they can immediately start taking that cell hostage, and they can work directly with the host replication machinery. If they're RNA viruses, well, they need an RNA-dependent RNA polymerase in order to make more of their RNA. It's just a way to make more copies of their RNA, because every RNA virus needs its own special copy of RNA. And then those retroviruses that are so funky, they use that reverse transcriptase in order to take their RNA DNA off of that. And so it's similar to, we've already talked about this. It's just a little bit more complicated because now we have the nucleus where some steps happen there. And so got to use the ribosomes in order to make the proteins. And they have to do that in the cytoplasm. And then the assembly, this is kind of weird. I always thought the assembly is always in the cytoplasm. Not necessarily. They're finding some viruses can actually do assembly back in the nucleus. So even though they make all their proteins out in the cytoplasm, they may ship them over to the nucleus. So basically, it just depends on what virus is being studied, whether or not assembly is happening in the cytoplasm or the nucleus. And again, logistical issues, getting everything in the same place in the cell, and then getting everything to assemble. And then when they go to, if they are enveloped, they have that membrane around them. It can be the cell membrane, but it can be one of the internal organelles membranes instead. So it doesn't have to be the cell membrane. Whoops, what happened there? Okay, so complicated, but obviously there's plenty of viruses that have figured it out. After they're assembled, they have to then get out, and again, very often it's lysis. So when you get, a sore throat from a cold virus, the sore throat is primarily due to the virus is successfully lysing out of your cells. So you get damaged cells and you start getting that sore throat. So that you can often feel. The other idea is they call it budding. What happens is they'll slowly come out of the cell. And as they're doing that, they do it slow enough where they have time to wrap part of that membrane around themselves. So the budding process, again, it depends. It can either be organelle membrane, cell membrane. So here they're trying to show this is the papillomavirus, which can cause HPV. Whoops. And here you can see they're trying to show the logistics of, okay, so they're getting in. You can see it's the entire viral particle that got in, and then the capsid has to break open or uncoat, get the nucleic acid into the nucleus, then go through transcription, but they're also showing both. They're showing transcription going on to get your RNA off onto this trajectory. And then they're also showing, yep, you got to make more DNA too. They're trying to show both. But then once you get that messenger RNA, the messenger RNA will get processed a little bit more in the nucleus, and then it gets out, and then it has to go through, find a ribosome, go through translation to make all the proteins. And then in this case, they're showing the proteins go back into the nucleus, get assembled in there. So and then they're trying to show the release step. So this one's a double-stranded DNA virus, so it's pretty quick. Once it gets in, it's pretty quick to take advantage of everything. Here's the pathway of the herpes virus. Whether it's oral or genital herpes, much the same way. And again, they're just trying to show the complexities of the nucleus versus cytoplasm. Looks like the nucleus is being used again for assembly. And then they're budding out by taking some membrane with them. HIV. The retrovirus idea. Showing the presence of the reverse transcriptase right there. Looks like they're assembling out the cytoplasm for this one. And then budding out, capturing some of the membrane with it. So again, it just depends what virus you're thinking of, how they're going to work through this in the animal host. But compared to phage, it's slower and more complicated. Plants are a little different. If you think about viruses that can infect plants, they don't seem, at least so far, they haven't found a virus that docks into a receptor protein. So that's very, very different. What they end up seeing is that the virus has to get in either through a wound or through a vector, some kind of insect typically, that will be feeding on, the insect will get onto the plant, start feeding on it, and then the virus is carried in the digestive tract of the insect. And as they're feeding on it, they will have some of the viral particles in their saliva, and it gets into the wound. So it's mechanical transmission. And then, so damage, animal vector like an insect. And then once it's in the plant, it can get into the next generation if the virus makes it into the seed. So as the seed is developing, if the virus gets in there, then of course the plant that germinates from that seed is going to get it. So very, very different in terms of they don't have that first attachment phase. They don't do that. So plant viruses are interesting. They can cause a lot of damage and economic loss for sure when you're trying to grow certain plants. However, they've also been known to be coveted. And so this tulip that you're seeing here, this striping pattern, that used to be, well, it still is, it's coveted. A lot of people really, really like that. They value that highly. Back during the time period of tulip mania, there actually was a time period over in the Netherlands where it was like this craze for tulips and very rare tulips. And this was one of them. If you can prove that your bulbs would produce tulips, flowers like that, you could sell those bulbs for a tremendous amount of money and you could get rich instantly. It was a really crazy time of history. It eventually collapsed and everything kind of went sideways. But for a while there, it was called tulip mania. They didn't know that was a virus, but it was a virus. And that's why it was so coveted, because it was rare and they didn't understand, how that happens. Now we know how to make that happen if we want to, because we understand the viral pathway. But so yes, they can cause a lot of damage, but on the other hand, they are also showing some beautiful patterns that can be highly prized, and you can pay, good money for that. So here they're showing two plant cells in this image and two neighboring plant cells. And what they're trying to show is that when you look at plant cells that neighbor each other, they do have these little tunnels between them. They're called plasmodesmata. Plasmodesmata. And so these are tunnels, essentially, all open, that allow for flow of things from cell to cell. Nothing huge, but certainly small chemicals, small proteins, viruses, sure, because they're small and all of that. And so it is, once they get in, if an insect is chewing and injects the virus in that way, then once they get in, they can go through these plasmodesmata from cell to cell to cell to cell. And so they can cause a lot of wide damage through the plant body. And it's all open. It's lined with a membrane, but it's all open. So it's just very easy for them to flow from cell to cell. And so that can cause devastating damage pretty quickly. And then plants have a vascular system. We have arteries and veins. They have analogous structures. And once viruses get into the vascular system, that's another way for things, for the virus to really move all over the plant body. So here they're showing the columavirus. And I'm not going to get into asking details on this, but just trying to show the process here. And then they're finding an aphid insect fits into that piercing. Aphids have a very piercing mouthpiece that they can use. And they're showing replication, assembly, and then the release step is really going through the plasmodesmata to the next cell. So kind of a different beast, a different method when you get into the plant world. All right, so with the, we're not certainly at total, we have ways of fighting back. And so genetic resistance, certainly, we can have this ability to adapt. We have an incredibly sophisticated immune system, as long as we have good preventative health and stay healthy. And so we have a variety of mechanisms in play that can help defend ourselves. And if you take immunology, you'll learn all about that. There's also this idea of RNA interference that can be used in order to mess with the viral particles so that they are thwarted from being able to finish their replication. And they're looking at phage therapy, where they're actually going to use phages to deliver different drugs. So yes, there's plenty of viruses that can be very damaging and deadly, but there are also some very promising things or ways in which we can use viruses to improve. So with phages, what's so good about that is they're bacteriophages, so they don't harm our cells. And so if we were to take in a phage that is engineered to carry an antibiotic or some kind of chemical that's needed, maybe it's a tumor killing chemical. They're looking at that too for cancer tumor therapy using phages. So the phage itself won't harm us. It's just a delivery vehicle. And so that's another thing they're looking at. So there's a lot of research going on with viruses. And, of course, we hear a lot about the ones that are causing problems, and of course, with COVID-19, but there are plenty of other good reasons to think about viruses and to study them and to benefit us. So I want to get into that slide set. I don't know if that's in EDUCAT. Does it mean the second part 2? Okay, let me put that. I'm gonna be updating part 2. I'm adding into it. I just haven't finished that yet. I'm reading some resource where they've made some really good progress with some of the viral ecology stuff. So I wanted to add in a little bit more, but I haven't finished it yet. So that the second slide set, I guess, calling it viral ecology, I guess. So this slide set, trying to show a little bit more about, just like with all the microbes that exist on this planet, the vast majority could care anything about us. They don't care. Same with viruses. They're not out to kill us. There's only a tiny, tiny percentage. Is it showing up now if you refresh? Okay. There's only a tiny percentage that are capable of doing damage to us or killing us. So, of course they get the limelight, but I'm hoping after you learn about this section of class that you'll appreciate that all viruses aren't evil and we actually need them. And they have been a very important part of the evolution of life on Earth. And they're actually very important for the human evolution too. So when you look at some of the jargon that they use when you get into this type of field of study, they talk about the virome or virome. And so it's just all the different viruses in that particular habitat that you're studying, kind of like the microbiome, virome. And so, you can think about this idea that, okay, sure, we know that some viral DNA can paste into the host DNA. Phages that infect bacteria can do that. So can so if you call it a prophage, that's specific for the viruses that attack bacteria, all right? And they've inserted their DNA into the bacterial DNA. If you say provirus, it just means it's the same strategy with an animal DNA. So it's an animal virus that's pasted its DNA into the host DNA. Then you say it's a provirus, okay? And again, we have lots of proviruses in our DNA. If you get your DNA sequenced, you would see a lot of stretches of proviral DNA in your DNA. And that's normal. That's just normal. That's the way we are. And so endogenous viruses are the term where you see the prophages, or more often proviruses, that it's just like always in the DNA. It doesn't ever seem to cut out. And it's actually like part of the host DNA now. And so permanently integrated. And, of course, that just means too, when they say transmitted to the germline, it just means that it can get passed down into any progeny, if you have kids. So they're finding that can actually be a good thing. It can provide useful traits to the host. They can see, for example, with the nitrogen fixation going on, so cyanobacteria are good at doing nitrogen fixation, and it's because of a prophage in their cells. That's how they think that this ability to fix nitrogen came into play. It's actually through that process. And so that's a huge trait. I mean, if you are a cyanobacteria like Nostoc and you can fix nitrogen, that's a huge advantage for you. And so the fact that they acquired that through this prophage idea, that's kind of like a permanent thing. That viral DNA just stays right in. So here's another thing that many people don't realize. As much as half the human genome may have arisen from viruses or virus-like elements. And so I don't know, do they talk at all about that in genetics class or evolution class? You might talk about transposons. You might have heard that term, but yeah, there's a lot of interesting work going on with the structures of jumping genes. You may have heard that phrase, jumping genes. And so when they start looking more carefully at the human genome, they can see, they call them virus-like elements, but viral DNA is there in the human genome. And so I'll talk a little bit more about that. The other thing that a lot of people don't realize, and I don't know how much it's talked about in our evolution class here, is how much viruses play a role in the process of evolution and the roles they play in different ecosystems, and that they really shouldn't be ignored. They've been making really good progress with research in ocean systems. So they're finding there can be 1 times 10 to the 7. So that's 1 followed by 7 zeros of viral particles in 1 milliliter of seawater. 1 milliliter. That's a milliliter. So, of course, that begs the question: what are they doing there? You know, that's a lot, and more so than what we find with bacteria in the ocean. There's more viral particles than bacteria. So it's really getting a lot of scientists to look hard at what's going on here. And so here they're showing this idea that you have both the phage type viruses that infect bacteria, the animal viruses, and you have both the infection process. Then when also they lyse open the bacteria, you'll have the disintegration, or the animal cells, the disintegration of themselves, release more. And so this idea of kind of looking at the turnover, I guess, of viral particles and how they end up in these different portions of these different habitats. So they're looking very carefully at that, and they're also asking about this with regard to like the coral bleaching idea. They're looking at, are viruses playing any role in that, in the coral bleaching and the coral death going on, or is it the reverse? Are they helping get things to stay healthy? And so there's a lot of work with that, looking at the idea here. So here again, they're trying to show the viruses are playing a role in, you always have to have nutrient cycling going on in these different habitats and on Earth in general. And so here they're showing this idea that the viruses, by doing their thing of infecting and replicating and then lysing out, that they are really intimate in the process of nutrient recycling going on, and the carbon balance in marine systems. This is the system they studied the heaviest, but they're suspecting this to be the case in other ecosystems too. So the viral ecology idea is really, this research is really getting some interesting results especially with the ocean systems. Okay, I think I'm going to stop there. The other things I'm getting into are a little bit longer topics, so we'll continue on with this on Friday.